Hello. చేస్తాను అందరిని అడ్మిట్ కొడుతున్నాను. మేడం ఆవిడ
Another.
Hello, Sridhar sir, am I audible?
Okay.
Sridhar sir? Sridhar sir?
హలో.
Sridhar sir, am I audible?
ఎస్ మేడం, ఎస్ మేడం.
ఓకే సార్, థ్యాంక్ యూ.
ఇచ్చాను, మైక్ కొంచెం లోగా ఉన్నట్టుంది వాయిస్. మేడం
సార్ చెప్పండి. సార్, am I audible?
ఎస్ మేడం, ఓకే. నౌ ఇట్స్ ఓకే మేడం.
ఓకే, ఓకే సార్, థ్యాంక్ యూ. A very good afternoon to one and all. I am Mrs. Sivajyoti, heading Department of Pharmaceutical Biotechnology, Hindu College of Pharmacy, Guntur. As the convener of today's program, it gives me an immense pleasure to greet you all here. A warm welcome to distinguished luminaries, delegates, faculty members, and students fraternity from various colleges and universities for inauguration of a 4-day international webinar, Conclave, on an intellectual war against the COVID-19 pandemic, being organized by Department of Pharmaceutical Biotechnology from 19th June to 22nd June 2020. I accord a genial welcome to today's chief guest, Dr. K.R.S. Samsivaravu Garu, Vice Chancellor, Mizoram University. I extend my hearty welcome to today's speaker, Dr. Anna Tels Ma'am, Postdoctoral Researcher, Germany. A hearty welcome to Professor K. Srinivasulu Sir and Dr. Amar Verma Sir. We welcome you, sir. Firstly, I would like to thank today's eminent personalities, Dr. K.R.S. Samsivaravu Garu, Dr. Anna Tels Ma'am, Professor K. Srinivasulu Garu, and Dr. Amar Verma Garu, who have accepted our invitation for this webinar to enlighten us. Hindu College of Pharmacy was established in 1999, located in the heart of Guntur, with the fervent desire to train the students as better builders of the nation and to inculcate skills of leadership in all aspects of life. The institution has grown substantially with 5 specializations in M-Pharmacy, 6 years PharmD and PharmD post-baccalaureate. Hindu College of Pharmacy was recognized as a research center in 3 departments. Our institution has a potential, dedicated, highly qualified, experienced senior faculty members with a zeal to encourage research activities among students. We have highly sophisticated laboratories, spacious classrooms, digital library, and computer lab. Let us start today's session with lighting of lamp. I request our technical team to start.
Kurutvam kalyanam arogyam dhanasampada shubham kurutvam kalyanam arogyam dhanasampada shatru buddhi vinashaya deepajyotir namostute deepajyotir namostute shubham kurutvam kalyanam arogyam dhanasampada shubham kurutvam kalyanam arogyam dhanasampada.
The main objective of today's webinar is to acquire in-depth knowledge on evolution and pathogenicity of COVID-19 and how to start biotech industries in the present scenario. In this regard, we have invited experts from various fields. In this webinar on day 1, we have the session on COVID-19, evolution and host immunity by Dr. Anna Tels Ma'am, postdoctoral researcher from Germany. On day 2, we have the session on COVID-19 evolution and pathogenicity by Professor K. Srinivasulu Garu from KL University, Vadeshwaram. On day 3, we have the session on host T cells and microRNA role in COVID-19 by Dr. Amar Verma Garu, postdoctoral researcher from Germany. On day 4, we have the session on innovation and incubation in biotech and pharma sector by Dr. Jahurullah Mohammed Sir, Chief Innovative Officer, Government of India. I request all the participants to utilize this opportunity to acquire knowledge on COVID-19. Now I request an able administrator, social activist, leading advocate in Guntur city, our chairman sir, Sri Jupudi Rangaraju Garu, to address the gathering. Over to chairman sir.
Good afternoon to all the persons for participating in this conference or meeting. I deem it a privilege to talk to you on this good occasion. Our Hindu College of Pharmacy is established with the object of imparting good education to the pharma students. Also got good library. And about 1 year back, we have started a research center also, and Acharya has recognized us as one of the research centers. We are striving for establishing a testing lab, and our institutions are more than 160 years old. We have got chain of institutions which are 12 in number. Our society comprising of doctors, advocates, chartered accountants, businessmen, who are all established in life, and we are doing only time for service education. Our Hindu College of Pharmacy has got good faculty, and we are imparting education in almost all faculty of pharmacy. And present situation of COVID-19, which is creating a havoc in the minds of the entire human society. Our college has taken object of in respect of this COVID-19, and I really appreciate my good staff who are genius and curious for bringing up for 3 to 4 days, especially persons like Mr. K.R.S. who is renowned and known to all persons in this part, and doing a generous responsibility in the Northeast. I also thank participants of students to get equipped with this COVID-19, and it is time that all the colleges members of the great fraternity should try to evolve a situation where if India can make a preventive drug or a drug to eliminate this COVID-19, so that we will be the champions of this and we will be the leaders of the world also. I hope that all the experts who are their knowledge and experience would cater to the generation in respect of such COVID-19 situations. And our Hindu College of Pharmacy has taken this good job. I appreciate all of them, and I especially thank Ms. K.R. Samcharagaru, and other allied members of this great fraternity for giving up their time, putting up time and putting their valuable support for giving good education to all the persons and all the children. And I pray to Lord Almighty that this COVID-19 should run away from the universe, so that all human beings will have a human life, not a human life. We don't want a human life, but we want a human life, so that there will be good connectivity in respect of human thoughts. I thank the organizers of this meeting wholeheartedly, and I pray to Bhagavati that all happy, and especially I put my thoughts that there shall be an endeavor from this meeting, that something concrete, at least to certain extent, would be made. And I want to put all my thanks to all the persons who are participating. Thank you. Bharat Mata ki Jai.
Thank you, sir. Now I request young, dynamic and driving force of our college, none other than our principal sir, Dr. R. Govindarajan Garu, to address the today's gathering. Over to principal sir.
Good afternoon to all pharma fraternity. On behalf of Hindu College of Pharmacy and our management, a warm welcome to all for first international webinar in our college. So this platform, I request the present situations, the COVID-19, what is the concept behind how they are occurring, what is the problem for the giving therapeutics. So all kind of your detailed deliberations will be giving this webinar. We are invited various stalwarts who are working on COVID-19 from different country. So in this webinar, please take attentions. Please raise any queries related to the COVID-19 issues. So in these junctions, I really congratulate the Department of Biotechnology to do wonderful the sessions, and also congratulate all the participants who are taking involved in the webinar for the make success. So thank you. Now over to HOD in the department, Dr. Sivajyoti.
Sir, international speaker wait చేస్తున్నారు. Madam Director.
Thank you, sir. Now I request intellectual, charming and cheerful personality of our college, our Vice Principal Madam, Dr. A. Sunita Garu, to address the gathering. Over to Sunita, ma'am.
Yeah.
Ma'am.
Good afternoon, ladies and gentlemen, dignitaries, invited guests, faculty members, my dear students. We keep moving forward, opening new doors and doing new things, because curiosity keeps leading us down the path. Thank you for joining us for today's webinar. With all my affable greetings, I cordially welcome you all. Hello. So, with all my affable greetings, I cordially welcome you all for International Webinar Bio Conclave. Your presence and patience makes us feel happy. Moreover, it is encouraging us to organize such online programs again and again. Here comes different departments of Hindu College of Pharmacy with a spectacular webinar all this month. We are immensely blessed to have the KRS Samsarao Garu, Vice Chancellor Mizoram University, as our today's chief guest. Your score of records, certificate of merits in various fields reveal your intellectual capacity. We do have a great sense of admiration and appreciation for your scholastic achievements, engendered very active involvements and leaderships in countless presence colleges, professional organizations, and universities across the world. Certainly, your presence amidst us today adds more joy and color to this program. We salute you with gratitude and extend you a hearty welcome. I cordially welcome today's speaker, Dr. Anatolis, postdoctoral researcher, Germany, our eminent personalities, Dr. K. Srinivasalu, Professor Biotechnology, Dr. Amar Verma, Dr. Jawaharlal, Government of India for this inaugural session. It's a great pleasure of mine to share with you on this occasion. The webinar will keep the young brain sharp and up to date. It's a time to nurture the hidden skills and talents of my fellow teachers. During this pandemic situation, Hindu College of Pharmacy in making the transmission of knowledge from the experts to the youngsters. Apart from the academics, the members of Hindu College of Pharmacy has rendered their services to the society. After the announcement of nationwide lockdown due to COVID-19 pandemic, this has thrown the livelihoods of lakhs into a chaotic twister. Daily wagers, footpath vendors, migrant laborers have all had their only source of income snuffed out. Don't worry. To help them stay afloat, the management of Hindu College of Pharmacy has stepped in, doing their bit in multiple ways. Hindu College of Pharmacy team donated 2 lakhs towards PM CARES fund. Not only that, our management came forward to provide food to daily wagers, homeless, and other people affected by the curfew imposed in Andhra Pradesh state in view of the coronavirus pandemic. Our team's mission is simple. No one should have to sleep hungry.
సార్ మిమ్మల్ని ఇంట్రడ్యూస్ చేయాలి డైరెక్ట్ చెప్పేసేయండి లైక్ మ్యూట్ అయిందా సార్
హలో
మేడం మేడం
హలో హలో
శ్రీధర్ సార్ ఫోన్ చేయండి మేడం నన్ను
Now I take this privilege to present you a video clip of spectacular services done by our management.
కేఆర్ఎస్ మాట్లాడితే వాడికి ఇప్పటికే ఫోన్
రుచిస్తాన్నేడే నవశకం నినదిస్తా నిత్యం క్షణహితం నలు పెరుగని సేవే అభిమతం కష్టం ఏదైనా సమ్మతం of the people for the people by the people ప్రతినిధిగా this is me this is me this is me మనుష్యులందు నీ కథ మహర్షిలాగా సాగదా మనుష్యులందు నీ కథ మహర్షిలాగా సాగదా ఇదే కదా నీ కదా ముగింపులేనిదై సదా సాగదా ఇదే కదా నీ కదా ముగింపులేనిదై సదా సాగదా నిస్వార్థమెంత గొప్పదో ఈ పదము రుజువు కట్టదా సిరాలు లక్ష వంపదా చిరాక్షరాలు రాయదా నిసీధి ఎంత చిన్నదో నీ కంటి చూపు చెప్పదా నీలోని వెలుగు పంచగా విశాల నింగి చాలదా మనుష్యులందు నీ కథ
Hello. Good afternoon to all the participants. On behalf of Hindu College of Pharmacy, the students, chairman sir, secretary and correspondent, and all the participants from all over the globe, I invite Professor K.R.S. Amsarao Garu, a great intellectual and versatile personality in the field of biotechnology, the present Vice Chancellor of Mizoram University, located in Mizoram, India, who has given his kind consent to deliver inaugural speech to this international webinar. He is versatile in respect of his academic qualifications and various prestigious academic positions that he has occupied, research areas, authoring of the textbooks, etc. In his 35 years of academic research experience in the fields of environmental toxicology, physiology, environmental and aquaculture, biotechnology, fermentation technology, etc. Sir has given guidance, and 25 scholars were awarded with PhDs, and 7 scholars are undergoing their PhD to pursue under his esteemed guidance. Sir has published 241 research papers and 8 patents filed, and 23 generic scientific articles published, and 68 papers presented in symposia of national and international levels. And Sir also authored 15 textbooks and 10 textbooks especially written for Center for Distance Education as an editor. Born on 15th August of 1958, Sir also has offered his services as Pro-Chancellor of Acharya Nagarjuna University, and also Sir has acted as the in charge of Vice Chancellor for Acharya Nagarjuna University, and many other positions that he has occupied, like visiting scientist for many countries like Germany, USA, etc. And also chairman and member of Board of Studies in Acharya Nagarjuna University, Sri Venkateswara Institute of Medical Sciences, Rayalaseema University, etc. And visited many number of foreign universities to fulfill various assignments in USA, Australia, Nepal, Germany, and many other countries like China, South Korea, etc. And sir is also expert in the field of biochemistry, physiology, biotechnology, cellular microbiology, environmental sciences, aquaculture, environmental toxicology, fermentation technology, etc. And as a recognition to his scholastic excellency, Government of India, through University Grants Commission, through Department of Science and Technology, Department of Biotechnology, has sanctioned crores of funds to carry out his research projects for the benefit of the world. Sir also offers his services as editorial board member for many scientific journals and also as a reviewer for biotechnology and pharmacy journals across the globe. And in a nutshell, Sir is an epitome of scholastic personality to be emulated by the present day's research scholars, teachers, faculty members, and students. By thanking everyone for patient listening, I invite Professor KRS Samsirao Garu to deliver his inaugural speech on this wonderful occasion of BioConclave, a 4-day interventional organized by Department of Biotechnology, Hindu College of Pharmacy, Guntur, Andhra Pradesh, India. I welcome you, sir. Thank you.
Good afternoon, Nagabhushnam. I thank you very much for your very voluminous introduction. I thank all of you for preparing that one. First of all, let me thank and convey my respects to Jupiter Rangaraj Garu for his kindness in inviting me for this webinar. And also, I thank the principal, vice principal, and many Nagabhushan and other faculty members who invited me for this webinar. It is a very nice topic like intellectual war against COVID-19. This is everybody is talking about COVID-19, post-COVID educational changes, and everybody is mentioning about what are the situation, trade, business, but intellectual war against COVID-19 is one of the very revolutionary, very novel idea. How intellectual people, how to do and how to combat this particular one. As you know that the global outbreak of coronavirus is a disease, it has created unprecedented actually and challenges to the society. And many people are now, if you just see today's statistics, it is more than 85 lakhs people in the world that are suffering with this corona disease, and several thousands of people died. And even in India, it has been day by day increasing very well. And it has almost like a pandemic situation for the whole country and the whole world. And it is one of the biggest pandemic disease which occurred after the World War. And people consider that Second World War is one of the very that is a pandemic situation, and a big human loss has been there. And entire world is fighting almost like more than 60 to 70% of the whole world. Actually, almost 200 countries are fighting against this disease. And many scientists and industrialists are now in the world are gearing up their academic activities, particularly research activities, to bring some kind of medicine for this one, and also to develop the vaccine, and a lot of things are going on throughout the world. And even in India, in the beginning, when lockdown started, when whole country has been affected, and we are really suffering with the lack of masks, even sanitizers, and even personal protection equipments. And India was suffering. In many areas, there was no masks also available, sanitizers also not available. So now whole country within a period of 20 days, 30 days, they got self-sufficiency and self-reliance of India, we can say. And now masks are being that is synthesized and stitched at all the places, and even PPE equipment also not been developed in their own place, because lockdown has created so many things for the people. Lockdown has taught us so many things. And earlier, no mask actually been available in villages, but even in villages also now masks are being stitched, and 2-3 layers masks also being stitched, and even PPE equipment also being developed in many areas. This is what actually the whole intellectual people who are actually guiding the people to do all these things. Considering the various abnormalities of this disease, like neurological, pulmonary, neuromuscular, and respiratory and rehabilitation and cognitive complications. And it is one of the important disease that people are even sometimes psychologically depressed. And this is going to be a very crucial situation that instead of going for severe complications, if you just analyze certain of the health issues like heart attacks. In India in 2015, it was expected that more than 23 lakhs of people died because of heart attack. If we just compare the whole country, yes, it is only few thousands. It is 4,500, 8,000 people died. And in the last 6 months, and even though a lot of people are in the pandemic situation, but the death mortality rate is not that severe in particularly this one. People has to realize this one, and people should not be psychologically distressed for this one. And they have to be very well, no, this is not going to be anybody's help. People has to take their own measures for protecting themselves from the particular disease. And they have to be, only the important one is not to mix with the people and go with only mask and only maintain the distance. This is what actually, if we can maintain this one and take more water, and you will get a lot of relief from the particular disease. And if you just see in India's condition, more than 2.5%, it is just only 2.5% of death rate in India, if we just see this particular one. And if we take the heart attacks, it is much higher than that. And this is one of the biggest warfare now. And the whole country and the whole world is fighting about this one intellectually also. And needless to say that work of scientists worldwide is going on very well. And it is expected that the clinical trials going on and several things and vaccine trials and even the injections and also the control measures also going on throughout the world. And it is an excellent opportunity for us to find. You have to remember one. There are so many pandemic situations in the world earlier also, but it has took more than 10 years, 15 years, 20 years, even 50 years for getting the relief of those diseases. But now we are not going to take that much time. We are going to get only the vaccine or even the treatment measures, or maybe within 6 months or 1 year, we may expect this one. And it seems to be immense economic burden that is most important in the whole world. Actually, the India's contribution, export and imports, it is not going to be that much severe because only 2% of our exports actually affected, and 1.7% of imports are going to be affected. But whole world, even people say that China is also very safe, but China economy is going to be down. China's 40% of the production is going to be down. Why? Because the orders for China has been cancelled, and many people who are depending on China's technology, now they are cancelling because of this one. And this is what the people say, that their health-wise they are safe, but the country's economy has been gone down because of the world's pandemic situation. This is what very important one, and people has to, and scientists worldwide have to develop technology for this particular one. In all the countries, it is going on very collective approach, and people has to go with the collective approach, and everybody is working on this and to fight against this virus and to find out the vaccine, find out the control of this virus, and how self-isolation and distancing, how aggressive testing has to be performed. And people are in the recent yesterday I was seeing even in some of these small places also, they are trying to test this particular virus disease very efficiently at their level. And innovative measures such as antimicrobial masks and films, disinfection chambers, and face shields, disinfection sprays, and sanitizers are coming up. It is coming up from different places, or disinfection masks. And I saw in some of the places that the anti-microbial and antiviral compounds, which are actually taken out from the tribal ethnomedicines, and they are also using for coating the particular mask through nanomedical applications, and they are developing. And also in some of the areas, even in our university departments, they have developed two kinds of one jelly material which can swallow the viral particles and allow them to pass away from the gut and intestine analysis. And also they are also coating the various nanoparticles for the control of entry of the particular viral material, or inactivation of the particular material is also been tried. And it like that in many universities, people are trying in small university places also.
The most important one is people has to be very, very important that they should not be pandemic and they should not be also been depressive in this particular situation. And the first vaccine safety trials are also going on in human beings. It has been initiated in the March, and it is going to be very soon. It is going to come to a world for a particular usage. And treatment options, new clinical trials are being developed for so many products, and every day various drugs are being tried, and several options are being given by various countries. And now you have to remember the only positive signs. Don't remember about the negative signs. Or what, how best we are going to have. And if we just take one of the examples of our state, we have more than 140 cases in our state now in the last 1 week. What is the advantage is, out of 140 cases, no one has gone to bed. All are actually asymptomatic positive patients, and they are having a viral infection, and they are actually not been bedridden, and no treatment only is been given, only just normal some kind of paracetamol and other things they have been using, and almost 40% of the people got recovered. Out of 140, out of 40% people got recovered. And the people has to understand this situation. If you are taking appropriate measures, there will not be any pandemic situations. And the individuals who are actually recovered, they are also sometimes their social disaggregation is also being taken place for many people. You have to understand that that is not going to be any problem once they are proved that they are negative for the particular cases. They don't need to be discriminated from other people. This is most important. And we are trying with that is Ayurvedic medicines, homeopathic medicine. Homeopathy is giving preventive measures for particular medicine, and we are all trying all these measures. And our majorly very important is Indians actually has got more immunity, and it is also going to help them. And people has to only psychologically whatever come and come whatever it is, we need to be very careful, and we need to be, even if it is coming, people should be very that is without any fear. They should be ready to face that situation. So this is my take home message is that it is intellectual war is going on against this COVID-19, and the outbreak of coronavirus is maybe because of various reasons, and many organizations are working throughout the world, and there is going to be a coordinated way. And several universities in Europe, North America, and Australia, and they are working for this particular development of vaccines and also treatment measures. There is nothing to worry about. This online mode is there, and even in education, we are now trying. Earlier, nobody bothered about online education, even though many things are available in online. But whole world now going on online, and several of the universities in the world actually they cancelled classes up to 2021, and even virtual demonstration of the practicals also going on, and whole world is now going on online. So the development came in different forms, even though during the post-COVID condition, of course, the immediate things which are affected is economy of the countries, and that can be adjusted very easily because the self-reliance slogan of our honorable Prime Minister. So we have to develop our own resources, but particularly the people, migrant labor, and they are going back to their homes. What they're not being workless. So what is that it is to be done? And what many industries are being closed. So what is that it is going to be? That is actually the skill development for the local area requirements, and people has to be trained in such a way, and local reliance, self-reliance has to be provided. That is most important. And the whole world is working towards the development of organizations contribute to scientific and expertise, advisors, and everything to combat this particular disease. The disease seems to be discriminated between populations in terms of death rate and virulence factor, and also seems to be vary between geographical region. In India, it is death rate is less. In America, death rate is high. Italy, death rate is high. But these are the reasons you have to analyze, and not to be pandemic. And we are facing challenges globally. Difficulties will arise. We have to come out with victorious things, and we need to be very collectively to face the situation. And particularly students has to be very important that nothing will happen to you. You learn whatever it is possible through online, and whatever it is possible, and the technical skills you can learn even after the situation comes to rescue, and you can go for practical training outside. and you can go for learning in the industries, and everything will be very normal very soon in a few months. But this has taught us we are going to develop a scientific revolution, intellectual revolution. That's why I'm calling this title of the seminar is very good and very motivating for us. And I hope that my introductory remarks, because I'm not asked for any scientific one. I'm just generally I'm talking whatever is coming to me, and I hope that it has been covered and nothing to be pandemic and everything will be set right within few months. But maybe another 6 months it may take. But you have to be very careful in adjusting with the online teaching mode, and the vaccines and also the treatment medicines will be coming very soon within 6 month period, and whole world will be rescued very hopefully, and we all hope for the best of the world. Thank you very much.
Thank you, sir, for your valuable information on this COVID-19 and your opinion on our today's webinar. Thank you very much, sir, once again. Thank you, sir. Now I request Dr. Amar Varma Garu to introduce today's speaker, Dr. Anatelis ma'am. Over to Amar, sir. I request our technical staff to connect to Amar Varma, sir.
Yeah, now connected. Can you hear me?
Yeah, yes.
Yeah, sir. Yes, sir.
Yes, sir.
Okay, so then
Sir,
I wait
sir.
for.
Yeah. I'm sorry for Amar Varma, sir. And also, I extremely sorry to be our today's speaker, Dr. Anna Tales. She has a very busy schedule. Even though just a small call, she accepted our invitation. Thanks once again. Very thanks from our Hindu College of Pharmacy, madam. Sorry for the interruption, madam. Because of your kind-hearted for the education of the student. So sorry for this event, madam. Now our Amarendra Varma sir, please give introduction on our today's speaker. Thank you.
Yeah. Could you please mute all of them?
Yeah.
So that I can speak.
Yes.
And introduce.
Yes.
Okay, I think Dr. Anatelis. Okay, so good morning. So it's my great pleasure to invite our guest speaker, Dr. Anatelis. So Dr. Anatelis did her diploma in biology from University of Evora, Portugal. She then got admission in a PhD program in University of Coimbra, Portugal. In this program allows her to do PhD thesis wherever she chooses to do. So therefore, she joined in Charité Berlin, Germany. This Charité, which is one of the largest medical universities in Europe. In that institute, she did her PhD thesis on immunology of reproduction, where she has investigated the role of maternal regulatory T cells during pregnancy. And she has contributed several research findings towards in her thesis. Some of them are how regulatory T cells control uterine microenvironment and how they facilitate embryo implantation, and some other research findings that came out from her PhD thesis are the origin of regulatory T cells during pregnancy and how they can be baby's best friends, regulatory T cells. And later, her group moved to University Clinic Magdeburg, Germany, where she did a brief postdoctoral research. And then she moved to Max Planck Institute of Evolutionary Biology, located at Cologne, Germany. So I think many of you know that Max Planck Institute is a premier research institute in Germany. And she's currently performing her research on immunogenomics and the evolution of the immune system. And today she would talk about the role of the evolution of COVID-19 virus and how host immunity combats coronaviruses. Now I welcome Dr. Anatelis to take over. Hello? Hello?
Hello, sir, thank you, sir. Thank you for introducing today's speaker, Dr. Anna Tellis, ma'am. Ma'am, we welcome you, ma'am, for today's session. Sorry for the interruption from our side. It's getting delayed for you. Once again, we are saying, please excuse us and sorry for the delay. Okay, ma'am, please deliver your lecture.
Um,
Or to Anna Tellis, ma'am.
Yeah, yeah. Could you please wait? So I think there is some technical difficulty. I think please wait for some time. I think there is some technical difficulty to share the screen, the presentation screens. Just a moment. Hello, Dr. Anna Tellis. Yeah, right. I think. Yeah, I think it's working. Anna, can you maximize it somehow? Yeah, it's working. Yeah, now it's working. Hello, Anna. Anna, can you hear us? Hello. Yeah, I think she's in. Yeah. Hello, Anna. I think. Yeah, I think. Yeah. No, Anna, can you hear us? Somebody please unmute Dr. Tellis. Somebody has to unmute. Technical team, please unmute. Yeah. I think Anna, no, cannot hear. So are you able to hear us, Anna? Hello? Can you hear us?
I hear.
Yeah, now it's okay.
You hear me?
Yeah,
Okay.
now it's
So
okay.
I think it was the microphone was not well set.
Okay.
I didn't realize.
Okay. Yeah.
So
were
I'm
you there?
sorry.
So yeah, sorry for us from our side as well. So it took some time. Yeah.
No,
So were you there
problem.
when I was introducing you?
Yeah, I hear everything. You were not
Okay.
hearing me, so I think
Yeah.
it was something with my microphone.
Yeah. All right.
Right. So thanks a lot for this opportunity. I'm very happy to share this information with you, and I hope that we can all learn something. Yeah, like there is a lot of new information coming every day, so might be that I'm telling you something today, and tomorrow we have new information regarding it. But it's already exciting to know these things. So I'll start to show you the coronavirus picture. This is like a cell infected by the coronavirus. All these yellow dots are a virus that are trying to enter the cell, and the cell is already in apoptosis, already going to death. The coronavirus name comes from a crown of spike proteins that are in the surface. And these spike proteins are the ones that are responsible for the entrance of the virus into our cells. The virus is also composed by a membrane of proteins and lipids, and this is the membrane that gets when we wash our hands. I see a line here that I don't know why is here, but I don't know. Maybe something from the program. So just a brief introduction how the virus enters the cells. It attaches to these ACE2 receptors that exist in the cells of the nose, mouth, or eyes. That's how the virus first enters the body, is through these membranes. And it attached to the angiotensin converting enzyme, which is this ACE2. After binding to this ACE2 receptor, the virus can fuse with the membrane of the host, with the membrane of the cell of the host. It's very slow. And after it fuses, the RNA, the viral RNA goes inside the host cell, where it gets replicated, and new copies of the virus are made inside the cell. As the infection goes, the cells start to produce new proteins of the virus and forms new copies of the complete coronavirus that then can encapsulate all the information inside and become a whole virus. And this goes on and on. So each cell can assemble millions of copies of the virus before dying, and the virus can then be released while the cell dies. And it can end up also in droplets from the lungs when we cough or sneeze that can escape, or even through the breath, right? Okay, so someone is painting in the slides, I think. I stopped listening. Are you listening to me? Working? The sound is working? Is the sound working? Yeah. So there are actually already 4 circulating human coronavirus that exist already in our population. They're already endemic, and they cause mild symptoms of a normal cold. This coronavirus were already evolved some time ago. The first one that was reported was a human coronavirus 229E that was discovered in 1960, and it is known to have diverged from an alpaca before this year. From the alpacas, it was transmitted to humans. Then there are other coronavirus, one that diverged from a cow around 890, another one that diverged from a bat in 2005, and another one also that diverged also from a bat between 500 to 800 years ago. These viruses are all, as I said, they're all in the population. They cause a common cold, so they are not really severe like the new one that we are facing now. But there were already other coronavirus that were giving potential severe symptoms, like the COVID-19 now. One of them was first discovered in 2002, 2003. There was like a big outbreak of severe acute respiratory syndrome. This was the SARS one, and it also diverged from a bat and was transmitted to humans by civet cats. This happened in Asia. And there was also another one that was known as MERS, so Middle East Respiratory Syndrome, that was diverging from a bat coronavirus before the 90s and transmitted to humans by camels. This outbreak happened in 2012. All these outbreaks were really severe, and they used to kill a really big percentage of the hosts. But they could stop the outbreak before it became a pandemic. But now we are facing a new coronavirus that is also similar to these ones, but less lethal, let's say. That arrived in 2019, as we all know, in China, and then spread all over the world, and now we can find it everywhere. So this was also diverged from a bat coronavirus and transmitted to humans by another host that is probably a pangolin, but I will talk a bit about it later. So all these coronavirus came from zoonotic origins, meaning they came from other animals that then find an intermediated host, and these hosts transmitted to humans. And the most common of the host seems to be a bat. And in fact, when we look to the proportion of zoonotic viruses in different mammals, we find that the viral richness is higher in bats, primates, and rodents. They have a lot of proportion of zoonotic virus, and also the total input of virus is also very high. You can also find it in other mammals, but these three big orders are the ones that have more virus. And also, when we look into the world map, we see that there are a very big number of viruses that are carried by both humans and bats. And this also tells us something about the history of many of these viruses that we share with the bats, maybe we don't share with other mammals. And some of the hotspots, I think, like for example, in sub-Saharan Africa, there are 16 of the viruses that are common between the humans and bats and that transmit diseases to humans. But there is almost no place on the world that there are no common virus between us and other animals. So we see that bats might be an important piece of this story. So why bats? Bats transmit fatal diseases in people, but we don't know. We didn't know why bats seem healthy anyway, even if they are carrying the disease. They could carry even diseases like Ebola, SARS, Marburg, Nipah, rabies, MERS, SARS, and SARS-2. All of these are very lethal diseases in humans. Virus seem sorry, I'm listening something. I'm listening.
I request the technical support. I request the technical support, please.
There is some interference in the sound.
Yeah, ma'am. No, it's okay, ma'am.
So why bats are special? Bats have a relatively long lifespans, and they can cover long distance when feeding. And they also can commute and migrate, and they have a social roosting behavior. And this roosting behavior usually also in the middle of the human populations. So they are very near us. And this is also getting more and more of a problem because of the desertification and because of the disturbance in the habitats of the animals. So they tend to come more to the cities and to be more in contact with us. But there is also something in the bats that doesn't exist in other mammals, and that can also make them a special mammal. So they fly. They are the only mammals that fly. And that's very important in this story because when they fly, this ability of flying has a relation with the immunity they have. In which sense? So there were already studies that shown that bats have a higher expression of genes that code for proteins involved in DNA repair. Here in orange we have some of those, so they are more expressed than other mammals. And these can allow a quick repair of the DNA damage done by the viruses. So this firstly, in the evolutionary story of the bats, arrived because when they fly, there is a lot of destruction also of their own cells because of the heat and the flight. And they need to have a mechanism to repair the DNA very quick. Otherwise, they could not have this ability to even to fly. But this also allows them to repair the damage of the virus and to survive 2 loads of virus that other organisms would not survive. Also, during the flight, the metabolism of the bats increase by almost 20-fold, and the heartbeat increases like almost 2,000 times per minute. And the body temperature can rise to 40 degrees. And this is existing in almost all the species of bats, not only one. So it's like a common mechanism in the bats. And also, like when the mammals are sick, they have a mechanism of fever, and the fever, it increases the metabolic rate to allow the immune reactions to control the infections. So fever actually is like evolutionary traits that allows to combat infections more easily. But the bats don't have fever. They are always in constant fever because every time they fly, their temperature rises like if they have fever. So this allows them also to have favorable innate and adaptive immune responses that are associated with this high body temperature that is also associated with the fever in mammals. Some examples like enhanced innate immunity and also enhanced adaptive immunity. So many cytokines and suppressor activity can exist in the bats in a normal state. Also, the high metabolic rates during flight may facilitate the costs of raising the metabolism to activate an immune response, meaning that they save some resources. They don't need to use the resources to develop a fever. They are already having this. But then also means that the virus, for being able to survive in the bat's body, the virus need also to be very strong. They also needed to evolve so then they can coexist with the bats. So this means that the virus need to survive to these such hard conditions that exist in the bats. So imagine when they don't have these conditions. This means that they can proliferate and they can infect other mammals much easier than bats, because they already learned in the hard school. So they were like in a school that is very hard, that are the bats that have very harsh conditions for them. Once they find another host that is not which immune system is not so developed, then they can infect easily. Also, something special about the bats is that they can also, because they have an expansion of immune genes like the natural killer cell receptors, MHC class 1 and type 1 interferon, they go into a state of high antiviral effect, but that allows them to be tolerant to the virus. So they don't produce so much inflammation. They just tolerize the virus without producing an inflammation. Just they keep the virus in check, and they don't suffer with it. So yeah, they are a very good reservoir of virus. And then they can transmit to other hosts. So how this happens? The bats can have a direct transmission of virus by bat saliva, urine, feces, or blood. And sorry, I don't know who is drawing this. Is not me. Just not don't follow whatever is shown here. They can transfer the virus to intermediate hosts that's like wild animals or livestock that can be infected with bat-borne pathogens. And also another important vector are the blood-feeding arthropods. They can also bite these hosts and transmit the virus through bites to another host. And then there is also the environment itself, which can have a role in the direct transmission through the contamination of the habitats. So this is how the interspecies transmission happens. But how do the virus accomplish these interspecies jumps? The mechanism basically relies on the access and ability. So if the virus can reach the cell of this host, it's successful. And if the virus proteins recognize and bind to structures as receptors in the host, then they are also successful to jump to another species. So it has been discussed which is the intermediary host for the COVID-19, the SARS-CoV-2. And this study has compared the whole genome of the virus of the pangolin. They compared different species. They compared the virus whole genome of different species with each other to see which ones are clustering with what. So which one are more close to each other than others. And they found that the SARS-CoV-2 virus from the pangolin clusters together with the bat and the human one. The first one from China. So this already gives a hint that this could be an intermediary host. And additional reassurance also comes from the comparing the ACE receptor. So the ACE2 receptor, both from the pangolin and the human, is very similar, even more similar than the one from the bat that was the first host, meaning that the virus would adapt to the pangolin receptor and then could pass to the human one because the receptor would be similar, so we could also enter the human cells. There is also a furin recognition motif in the cleavage from the viral entry that would allow the virus to go inside, and this protein was more similar to between bats and pangolins, but the human one seems like diverged from this, means that is a new one that maybe gives the virus a new chance to develop in the human organism, so like a specification for the humans. So we all know a bit of the story of the virus, that the first clones were detected in China. This means here, I'm just showing you 3 of the evolutionary genomes of the COVID-2. So the first clone that was was sequenced was in December of last year, and it comes from Yunnan province. And we can see that this clone is still in the population until now. Well, these are data from May. I think they didn't do after that, as the cases in China lowered down, but it was active since December to May. And around January, February, there were new mutations that were related with the virus arriving in Europe. And this is like kind of European and cluster from European and started in Europe. But then, of course, as we can see by all the colors everywhere, so now we have clones of different sources everywhere. So it's like the virus is mixed everywhere in the world. We don't have, but we can trace it to the origin by analyzing the genome. I also took the same print, what you can see in India, and yeah, you also see that the clones that exist in India also come from the different the common ancestor was also the one from China, but then also diverged. So you have clones that came both from Europe or from China, from the US, and they are still present in the population. So as the virus itself also has different mutations that you can see when you do these clusters. It's based on mutations that the virus has. And this graph, you just can see that in some regions of the virus genome are more prone to diversity, so they're more prone to accumulate mutations than others. This probably has to do with the conformation of the gene itself, so when it's more available or is more, so like places where the mutations are more easy to occur. And as this diversity also leads to the evolution of the virus itself. So with accumulating mutations, the virus can become also different. It can become more infectious, can become less infectious, can gain different properties than the first one, the original one. So I want to talk a bit about the evolutionary arms race between the virus and its host. So this means that the virus evolved new mechanisms to overcome the host defenses, and by the other hand, the host immune system evolves also a way to fight the virus. So we have always, we call an arms race, that one evolves and the other one has to evolve as well. And one of these mutations, that one of these factors that leads to the evolution of the virus, or that makes it more effective or not, was found to be the variations in the ACE2 receptor. There were studies that show that missense variants of the ACE2 are predicted to encourage and inhibit interactions with the SARS-CoV-2 spike and contribute to a genetic risk in COVID-19. So different variants of ACE2 could be leading to a facilitated or a harder entrance of the virus in the cells. And these variants were found in different populations in the world. So some regions of the world were more associated with a variant that would allow the virus to enter easily, and some were are associated with the virus and to be more difficult to enter. And another thing that is pointed in these studies is that ACE2, it's present in the X chromosome in humans. So this could be a reason why males have more severe cases of COVID than women, because males only have one X chromosome. So all the variants that they would have are the ones that are in this X chromosome, but women have two X chromosomes, meaning that they have more variability. They could have two haplotypes and one could be more protective than the other. So they could have a mosaic that protect them against, regarding to these variations in the ACE2. Another mutation that they found that was increasing the virulence of the virus is a mutation called D614G, and is also a mutation in the spike protein that can increase the number of functional spikes to 4 or 5 times, and make the virus more able to enter the cells. So this would be a mutation that makes the virus be more infectious, and this is already circulating. Though they say that the variant is not more dangerous than any that is existing now. It's just it's fast. I'm sorry, I think it was a problem with the internet. I hope you are listening now. see anymore.
Yes, ma'am.
Yeah.
Yes, ma'am. You are audible, ma'am.
Okay, great.
Yes, ma'am. You are audible, ma'am.
Great. So one of both the viral spike protein and the human sodium channel in the human cells need cleavage by furin protein. And what they found is that the virus seems to be mimicking this furin cleavage site. And it's very similar. With the spike protein from the virus is similar to the cleavage site of the human sodium channel, meaning that they might compete because they both need furin and they are very similar in the binding site. So it might be that the furin is binding to the virus instead binding to the human sodium channel, and this causes disruption in the human sodium channel and also allows the virus to infect more cells. This was one mechanism that the virus found to use the resources of the host. So now going a bit into what's happening during the COVID infection, how the immune system deals with it. So first we have the virus that enters the cell and replicates, and then after replication and release of the virus, the cell dies through a mechanism of pyroptosis. And macrophages get activated and start secreting cytokines that then can communicate to other immune cells, like either T cells or monocytes, macrophages, that there is a positive feedback. They produce interferon gamma that then brings more cytokines that then would attract more immune cells, and it's like always can induce a cascade of cytokines. This is a very common process in the SARS-CoV infection, and it's also the process that leads to severity of the cases, because when the immune system is dysfunctional, this cascade of cytokines, it's increasing more and more. And there is a super inflammatory reaction in the lungs that leads to a lot of destruction of cells. So not only the virus is destroying the cells, but also the immune system reaction to it is destroying the cells. That's how the cells of the lungs get destroyed. And then in the end, there is pulmonary edema and pneumonia and widespread inflammation and multi-organ damage. This is mostly promoted by the immune cells, macrophages, monocytes, and T cells. But when we have a healthy immune system and a healthy immune response, we have the clearance of the virus by the macrophages in the lungs, and also the production of neutralizing antibodies that can bind and inactivate the virus. We have phagocytosis of the apoptotic cells, so then they are not promoting more of inflammation. And there is also the prime of CD4 T cells that mediate an efficient immune response, and CD8 T cells that also can recognize and eliminate infected cells. So in this case, there is a minimal inflammation and lung damage, and the infection can be controlled. So like focusing more on the T cell responses, there is this study that came out in Cell, I think last month, showing that both CD4 and CD8 T cells can have specific epitopes that can recognize different proteins of the virus. They can not only recognize the spike protein, but also the M, the N, and also other NSPS. And what was also very interesting is that unexposed individuals also show some degree of reactivity of CD4 and CD8 T cells that is proposed by these authors that is a product of the coronavirus that already exists in the population. So these ones that only give symptoms of a mild cold that are already endemic in our population might give some degree of recognition of the new coronavirus, because some epitopes are similar. So it might be that these memory T cells that already exist can recognize also the new coronavirus. And yeah, this is the last study I would like to talk about. It's a GWAS study that came up this week in NEGM. And in this study, they analyzed genome-wide associations in patients from Spain and Italy. And they found that there were 2 clusters that were associated with COVID respiratory failure. One of them is in chromosome 3 and has includes a lot of genes that are related with cytokines and that could be related with the innate immunity and also with lymphocyte homing. And another one, it's in the ABO group, blood type group. So they saw that individuals with blood type A are more prone to severity of coronavirus disease than type O. So type A have a higher percentage of severity compared with blood type O. And they think this is because type A blood has already more coagulation factors, which in this case is important because many of the implications of the corona are also happening in the circulation, and this could lead to the formation of thrombosis and also disruption of the endothelial cells. So yeah, this is what is found now. And yeah, this just to remember that ACE2 expression exists not only in the lungs, but exists in many organs in the body. So like ACE2 is in blue. As you can see, there is a lot of expression in the lungs, but also in the enterocytes, in the intestines, in the eyes, in the liver, in the heart, in the kidney, pancreas, and even the placenta. So meaning that the virus has the ability also to bind to these tissues. And when it also binds in the blood, then we have also the formation of thrombosis and disruption of blood vessels. And yeah, this can also be seen from different ways that our organs are affected. So the virus can also affect the kidneys and the liver and the intestines after it enters the body. So I should just like to finish that this is a new virus, as we all know, so many things are still unknown. This is information we gathered so far, but then there is still a lot to be understood. Many of these things are, for example, that we have many asymptomatic people that get the virus and don't have symptoms, but they can spread it. That's why we need these measures of social distance, because you never know when you are infected and you are spreading it. And it's still not really understood why some people have severe case and other not. Yeah, we have some information, but it's still a lot to be known. So then I would just like to thank you for your attention, and I'm happy to answer any questions.
Hello, ma'am. Thank you, ma'am. Thank you for your wonderful lecture, ma'am.
Thank you.
You have elaborated the molecular mimicry, healthy immune response, and targets of T cell responses. Very nice lecture, ma'am. Even I too, it's like a wonder for me. Very good knowledge we acquired today, ma'am. Thank you once again, ma'am. From our side, again, we are saying, please excuse us for your disruption in the morning, in the 1 hour before session. Okay, sorry, ma'am, for that. If anybody is ready to ask questions, we may start the questionnaire section, ma'am.
Yep. So,
Amar?
Yes, sir. Any questions from the students? See, anybody is raising their hands?
If any participant want to ask any question, please type in chat box, then we can unmute.
Any questions?
Yeah, all the participants regarding feedback. So we will send the feedback link along with the second day invitation link. Today we don't have sent any feedback link. So please patience. If you have any questions, you can type in our chat box. Okay. So it is a notice for you. All
Amar, I have a question.
Excuse me?
Amar, I have a question.
Yeah. So,
Yeah.
Doctor, so you can directly ask Dr. Anna Telis.
Anna, this is Professor Srinivasalu. Hello.
Yes.
Yeah,
Hello.
yeah. I have a question with me. So in Indian, that COVID strain is similar to
type 1 interference.
Yeah, similar to China or USA strain.
So according to the graph that I looked, it's similar to all the strains. So like they have China, Asian, and also well, I don't know if it's really China, but it's Asian, and also from the USA and from Europe. So all of the strains are there.
In India, they are telling that our strain is mutated. It is less pathogenic than USA. Is it true?
I don't really know. I don't really know. So I know that there are some mutations that are happening more in some populations than others. So might be that's the case that they already seen in India. I don't know specifically, but could be.
Nice. Thank you for your. Yeah.
Madam,
Hello.
hello.
Yeah,
Verma
good
sir.
evening.
Good evening, Verma sir, and good evening, Dr. Anna Telis. Your presentation is very excellent, madam. Then some of the technical issues may be drawing the your PPT, but next time we will rectify that one. Then some of the participants asking the question, why one of the participants asked one question, why males are more prone to the COVID-19 than females? What might be the reason? This is one of the
So they are
questions
trying to figure out that. So there are different hypotheses. One of them is that ACE2 receptor is present in the X chromosome. So as males only have one X chromosome, they only have one variant of the ACE2, but women can have two variants. So meaning that if one it's more prone to the infection or makes the infection easier, might be that the other one existing the other X chromosome, it's balancing that, so it makes it less infective. So they have more chances that it's not so infective. Another hypothesis is that sexual hormones have a role as well, so that can increase the inflammation and the inflammation reaction. And estrogens might be more balancing, more towards balancing, but this is still not really understood.
Thank you, madam.
So there is another question from someone that I think they are missed your last slide. So they are asking why blood group A people are more prone to infection.
So all of them are prone to infection. The thing is that the people with the group A might have more severe cases of COVID. So the disease can be more severe in the A type. And yeah, they still need to understand better that why is the reason, what's the mechanism behind it. But one of the hypotheses is because people with blood type A have, in general, since the birth, they have more coagulation factors than people from blood type O. And having more coagulation factors is good, for example, when someone gets wound and needs to recover, so the wound cicatrizes very easily, but they can also lead to the so it can be more easy to form thrombosis. And in the COVID disease, this is an important, also an important factor. Is also an important factor for severity, because the people also have a lot of thrombosis, and the disease is not only in the lungs but also in the periphery. So it might be that these coagulation factors have a role.
Yeah. So then there is someone asking a question regarding the suppression of immune response, I think. So what he's asking is, recently one of the drug named dexamethasone, I think, which is a corticosteroid, I guess, is I think, yeah, is used for COVID-19, but he's saying why, I don't know why, because just he has given a statement. So now I think I can interpret his question. So is it good to use corticosteroids during COVID-19 infection, given that there is more collateral damage from the side of immune system, so suppressing the immune response? So any ideas or thoughts?
Yeah, I don't know exactly. Yeah, but that's a good question. So if there is a role in the hormones, might be that is happening, and is also dampening the immune system, like using the use of corticosteroids can also make the immune system less inflammatory.
Yeah.
So if the problem is that there is too much inflammation, the use of corticosteroids might help. But I think there is still not enough studies to say or to advise someone to take it for the disease. Yeah, but it's an interesting question.
Yeah, all right. So any more questions?
Yeah, one question, one point is COVID-19 is a disease developed from is a microbial evolution, or it is generated from the Wuhan? The question asked by the one participant. It is a microbial.
So it's whether it is man-made or so that's the question.
Okay. So what all the research is showing is that it's a natural born virus that evolved in other hosts like the bats, and then was transmitted to intermediary hosts until it reaches the humans. Yeah, so this is what the research is showing, is that it's very similar to other viruses that exist in other animals. So it's not that it was produced in the lab. It's something existing in nature that is just passes and just evolves by itself. Other animals have it too, but maybe they don't have symptoms like us.
Exactly. So then I have a question on a perhaps it may provoke you, but before that I give a disclaimer that I am against this view. So there is some kind of conspiracy or concept or hypothesis going on that it's not actually a virus, but it's just simply an exosome which is passing. You know? So any thoughts about it? It's not actually a virus, but it's just an exosome, you know, budding out of the cells and host cells, and simply they are making story out of it. I am against it, because if it is the host-derived exosome, then there is no need for the exosome to target and destroy the host cell. That's my view.
Yeah, this would mean that it's like an autoimmune reaction, right? And that would exist already without any transmission. And what we see is that there is transmission. So
Yeah.
You can trace where the virus came from. You can trace the transmission from which person or from which clone it's coming. Not all the clones are also the same. So
yeah.
Another question, madam. How much time it will take to develop the vaccine? If you are getting the vaccine, it is work on the mutated strain or not? That is the question asked by the one participant.
If the vaccine will be effective against a mutated strain, let's say. So when they build vaccines, they look for conserved regions for conserved epitopes from the virus. So like this will be parts of the virus that don't mutate so easily. This is one of the. So a vaccine should be effective even if the virus is mutating. It's not like the influenza virus. The influenza virus is mutating really fast and it's a very short virus, so there is not much to build a vaccine on. But the coronavirus is a quite long virus and is more stable as well, so it doesn't mutate so fast. So the chances that the vaccine will work in the next year and the next year are very high. And they look really for these conservative regions, so that's also something that
I have another question, a layman question, ma'am. Yeah, in India, one proverb is there about the common cold, because this COVID is also sibling to the common cold virus, as you explained already. So if you take medicines, it will cure in 7 days. I'm telling about the common cold. Otherwise, it takes 1 week.
Mm-hmm.
So means as for that quote, there is no remedy for the viral like common cold. This will continue, the COVID also continue like that manner, because the death rate is very less in India also.
Mm-hmm.
So some are telling that only the corporates are creating fear. Actually, the death rate may be. I don't know. Yeah. Is it
the
true?
thing is, the problem is that we don't really know. We don't really know the consequences, because for knowing that someone is dying with the virus, you need to test.
Yeah.
And I don't know how are the tests in India, but in almost all the countries that I know, they test mostly for people that have severe symptoms.
Yeah.
And then they try to trace it from the people nearby if they have the virus, even if they don't have the symptoms. But might be that also many people die and they don't know that they die from the coronavirus. And yeah, if they are not tested, they don't know. But it's also true that there are a lot of asymptomatic people with no symptoms that have mild symptoms, and there is no problem. Maybe for most of the population, the problem is that these people without symptoms, they can transmit it to people to whom the virus will be lethal. And this virus is very associated with people of old age.
Yes.
And also people with diabetes, hypertension, and metabolic diseases. So yeah, that's the problem. So for a person that is healthy, usually there is no problem, but the problem is that this healthy person can transmit it to people that cannot handle the virus. So if the population is very old and has a lot of problems of metabolic diseases or diabetes, might be that the percentage of people that will die will be very high. I imagine that in India, the population is very young, and might be that the virus passes to the young people without any problem. But if they go to the home of their parents, maybe the parents will not handle it. So
thank you. Thank you.
Yeah. So one last question, if there is any. So
close, no problem.
Yeah. So then, so management. So where is management?
Yeah.
So yeah,
Yeah.
you can give closing remarks so that.
Yeah. So I take this opportunity to propose vote of thanks. So a big thank to Dr. Anna Tails, madam.
One small announcement. Total visitors of this total thing is around 2,270. 2,270,
No,
including
no,
you.
sir. No, sir. It is around 2,506, sir.
Oh, later increased. Yeah. Oh, great.
Yeah. So then they are more enlightened by your talk, madam.
It
Thanks
shows
for the
our
kind
population.
acceptance.
It shows our India population, madam. Directly proportional, please. Yeah.
So I think big thank to Dr. Anna Tails, madam, for her nice, wonderful talk and patiently give the answers to the question and answer. And the participants are enlightened with get the knowledge, acquired with the knowledge on the COVID-19. Thank you, madam. And
thank you.
Thanks to really thanks for your nice presentation, madam. Thank you.
Thank you.
And also, I thanks to the today's chief guest, Dr. KRS Samsarao Garu, for his wonderful speech on the COVID-19. Then I thanks to the management, Hindu College of Pharmacy, for their encouragement and support conducting such type of webinars. So I thank to our management, Dr. Chairman Sir Jupiter Rangaraj Garu and Secretary Dr. Masoon Rao Garu. Then I thanks to the Principal Sir, Dr. Govind Rajan Garu, for his support. I thanks to the Vice Principal Madam, Dr. Yesunta Garu, OSD Sir, Sitaramai Garu. And I thanks to the Navabhushan Garu for introducing the chief guest. And I thanks to all the head of the departments. Then faculty members, technical and supporting staff. Then I thanks to the particularly special thanks to the Amar Varma Garu and K. Srinivas Garu for your suggesting such a wonderful speaker on the present buzzing topic COVID-19. I hope our participants are enlightened with this talk. Thank you, madam, for his wonderful talk on this particular intellectual war against the COVID-19. Thank you once again to the Amarendra Varma Garu and Dr. K. Srinivasa Garu. On behalf of the department, I personally excuse me, madam, for your time delay and some of the technical problem. So I thanks you for your kind patience. Then with this, I hand over mic to our convener.
Yep. So I think Dr. Anna Tails, so I think you may leave. So I think thank you once again from my side for accepting my invitation without hesitation. Yeah. I'm
without
also sorry. Yeah.
Without Amar Varma involvement, it is not possible to deliver a guest lecture. Yeah. So it's only with the help of Amar only. Yeah. Thank you. We need to tell thank you to Amar Varma. Thank you.
So also thanks Anna. So there was some technical glitches. Someone is doing some silly things. So,
yeah, I see.
Yeah. So I think someone is more excited to say thank you, madam.
Yeah. Madam will leave now.
Thank you for the opportunity as well. It was very nice.
Yeah.
Thank you, ma'am. Thank you, Varma sir, and thank you, Srinivas sir.
Yeah.
And finally, I thank to all the participants for your kind support for today's session. Now it is the time to end the session. Before ending the session, regarding feedback form, I want to say one thing. We will send the feedback form for today's session with tomorrow's Zoom invitation. And we are going to close the session, ma'am. Thank you. Tomorrow we have the session regarding COVID-19 evolution and pathogenicity by exactly at 2:30 sharply by Dr. K. Srinivasulu sir from KL University, Vadeshwaram. Please all the participants be within the time. I request all the participants, special announcement is there. Please don't draw any unnecessary thing on the host screen because they are here for us to deliver valuable information. So we have to give the respect to the host speakers, everybody. So please cooperate and support us. Don't draw anything unnecessarily. Thank you for this. And I thank to all the participants and we are closing the session. Good day. Stay home, stay safe.